Hello everyone.
Hello, ma'am.
How are you, Risha, Anvadya?
Ma'am, I'm fine. How are you?
I'm also fine. So Shreya will be joining today's class or not? Do you have her number? Let me call her. Yeah, okay. So today we are going to start chapter number 13, that is photosynthesis in higher plant. I think this chapter is already started in your school, right?
Yes, ma'am. Yeah.
Okay. So please switch on your cameras, beta. How are you, Anvadya?
Good, ma'am. How are you?
I'm also good. So Risha, you reached your home?
Ma'am, we are in the native right now.
Okay. Okay. ठीक है, fine. Should I start the class, Asmi?
Yes,
Okay,
ma'am.
fine, beta. Okay, so what do you mean by photosynthesis? Can you give me a definition of photosynthesis? Anyone? Risha, Anvadya, Asmi.
Ma'am, it is a process of the plants to in the presence of carbon dioxide and water, with the help of carbon dioxide and water in the presence of light and chlorophyll to make food for itself. Yeah.
Yeah, it's the process of preparation of food by plants in presence of light with the help of carbon dioxide and water. Okay. So as a result of this, the carbohydrate, or we can say glucose or 6-carbon sugar, as well as oxygen are produced. So what's the food of plants is carbohydrate, or we can say glucose. So the concept of photosynthesis was unknown till the 19th century, but like a photosynthesis can be explained with the help of an equation. So this is carbon dioxide and water. They combine in presence of light to form CH2O, that represents a carbohydrate, that may be glucose or some like any 6-carbon sugar. But in end of, like we can say, 1980, 1897 to 1985, there is a scientist, you have to remember its name. He gave the concept of photosynthesis, and he was doing studies on purple and green bacteria, and he demonstrated that photosynthesis is a process that depends on the light, in which the hydrogen from any suitable oxidizable compound reduces the carbon dioxide to carbohydrate. This expression can be explained with the help of this two hydrogen ke atoms, and this A represents sulfur or any other compound that is that is used as a suitable oxidizable compound in presence of carbon dioxide, and light is responsible for formation of CH2O, that is like any carbohydrate, and water is released. So what happened? Mm, yeah, Do you have any idea about what is oxidation and what is reduction?
Yes, ma'am.
Yes, ma'am.
Okay, tell me. Risha, tell me.
Oxidation is like the addition of oxygen, and reduction is the removal of oxygen.
Is like that?
Yeah, we have learned
Sir,
that.
in ma'am, in chemistry, we have learned
Okay,
it's
so
oxidation is the addition of O2 and or removal of H2.
Yeah.
okay. That's for, I think that is for chemistry. In biology, oxidation and reduction means increase in number of electrons, and
And that's
oxidation means
another definition. Yeah.
yeah. So for here, for this like biology, we are going to use this definition, and like oxidation means decrease in number of electrons or removal of electrons. So reduction basically results in increase in number of electrons, and oxidation means decrease in number of electrons. Okay. So what happens in green plants? Water is the donor and is oxidized to oxygen. Here in this reaction, water is act as electron donor and is responsible, and it get oxidized to oxygen. So suppose if water is not present, then H2S, sulfuric acid, became a hydrogen donor for purple and green sulfur bacteria, and the oxidation product is sulfur or sulfate, depending on the organisms, not oxygen. So suppose if a question is given, okay, there is a reaction in which the water is acting as a hydrogen donor, then what will be the result? Then you have to answer the water get oxidized to form oxygen. But in case of purple and green sulfur bacteria, in place of water, H2S act as an hydrogen donor. Therefore, the oxidation of H2S results in formation of sulfur or sulfate instead of water. Okay? It's
Ma'am, can
fine?
you repeat?
Okay, fine. What happened in case of plants or green plants, water act as hydrogen donor and it get oxidized. Oxidized means the electrons are released. Okay, it will give electrons, and reduction means increase in electrons or gaining electrons. So the water get oxidized to sorry, the H2O get water get oxidized to form oxygen in case of green plants. But in case of purple and green sulfur bacteria, instead of water, H2S will act as a hydrogen donor. Therefore this H2S get oxidized to form the sulfur or sulfate according to the organisms instead of oxygen. Okay, during the process of photosynthesis. Now so what this scientist thought that oxygen is evolved by the green plants. uh, Oxygen evolved by the green plants come from water but not from carbon dioxide. This scientist thought like that. Okay, the oxygen that we are getting as a byproduct of photosynthesis, the oxygen that we are taking from plants for our respiration is coming from hydrogen but not from carbon dioxide that is involved in photosynthesis. And he proved the same with the help of an equation given here. So what he explained is 6 molecules of carbon dioxide combined with 12 molecules of water in presence of light to form C6H12O6. This is the glucose. And as a byproduct, 6 water molecules are again released and 6 oxygen molecules are released. This is the we can say this is the equation that represents, simple equation that represents photosynthesis. You must have studied this in earlier classes also.
Yes, ma'am.
Okay, ठीक है. So here C6H12O6 represents glucose and the oxygen is released from water. This was proved using the radioisotope technique. So here the oxygen released this from this 12H2O, the oxygen is released, and how how this oxygen is released from water was proven with the help of radioisotope techniques. That is of it's not of our interest, so I have not added much more about that. Okay. Okay, so it's clear till now. Should I proceed? Yeah, Risha, which one more chapter your ma'am has taught you in school?
That nervous system chapter, I think.
Okay. Just after, like after class, please let me know, है ना?
Yes,
Because
ma'am.
I have to. So after finishing this chapter, how much syllabus, like how much chapters are left for us? Because I want to finish your syllabus and I have to take objective test for all the chapters. So I'm planning for that also. So when your school is going to conduct final exams, is there any idea?
Sometime in
I
January.
Feb, Jan and Feb, yeah. What our class teacher told was February, but there are many teachers who are telling that it's January also. So we don't know.
But
Okay,
ma'am, January we'll have our pre-boards.
okay, fine. So how much chapters will be left so that I can plan accordingly? I think I have to take regular classes from now onwards so that our
I guess four more. Like
more,
after we, yeah, four more.
4 more.
Like excluding this chapter, we have respiration in plants.
Hmm?
1 minute. I just need to check if that's deleted. Just a minute.
Okay, ठीक है. After class, you can do this. Okay, just do give me a favor. After when you get whenever you get time, like after 8 o'clock or 9 o'clock, I don't know when you get free. Just send me the names of the chapter, and if there is any chapter which is like completely uh, present in your syllabus, then I will consider that first. Because the deleted portions, अगर नहीं भी पढ़ेंगे तो like for your exams, it will not create any good like 1 minute beta. Okay, so okay, Risha, I'm going to start class beta again. Right, resume. Where are you, Risha? I'm on mute. No. Okay. So where does like do you have any idea where did the photosynthesis takes place in plants? Asmi?
Leaves.
Yeah, leaves may be particular like
Green leaves.
ग्रीन लीव्स में क्लोरोप्लास्ट के अंदर या थाइलेकॉइड्स में क्लोरोप्लास्ट में भी थाइलेकॉइड्स में ओके सो photosynthesis takes place in the green leaves of plant but it also occur in the other green parts of plant except leaves. So what happen, uh, generally the chloroplast align themselves along the walls of mesophyll cells. So I told you that the leaves consist of mesophyll cells, palisade cells, upper epidermis, lower epidermis. Do you remember the structure, anatomical structure of leaf? I told you in one chapter. I don't know, like the name of chapter. I don't remember, but I have told you the like anatomy of leaf. In that, the mesophyll cells I have taught you. So these chloroplast align themselves along the walls of the mesophyll cells so that they get optimum quantity of incident light. So what is incident light? Incident light is the light that falls on a subject like chloroplast is subject here, either directly or indirectly. So the light that is falling on the chloroplast directly or indirectly is known as incident light. Chloroplast or any other organ of the plant. Okay. So yeah, within the chloroplast, there is membranous system consisting of grana, stroma lamellae, and the matrix. So we have already studied the structure of chloroplast in one chapter, right? Risha, where is Risha?
Yes, ma'am, I'm here.
हाँ बेटा, okay. Please on your camera, बेटा. Okay. So this is the structure of chloroplast. This is the outer membrane of chloroplast. These are grana that are present in stacks. So grana are the stacks. So particular एक portion of grana is known as stacks of grana is thylakoid or stacks of thylakoid is grana.
Stacks of grana is thylakoid.
Yes, ma'am.
Okay, fine. So, and this is stromatal lamella that connects to grana. This is the inner membrane and outer membrane. This is the stroma where all the cell organelles are present. These are the ribosomes. This is the starch granule and they are the lipid droplets. This is the structure of chloroplast. Okay, that we have studied in one previous class also. So these are the structures of chloroplast. So what happened? Like there is a clear division of work in each part of chloroplast. Like the grana of chloroplast is having an individual function. The stroma of chloroplast is having an individual function. So each portion of chloroplast have its own function. So there is a division of labor in chloroplast. Here the membrane system, this membrane system means outer membrane and inner membrane of chloroplast is responsible for trapping the light energy and also is responsible for synthesis of ATP and NADPH. So this is the function of chloroplast membrane. Okay. Now this is stroma. This stroma in chloroplast is responsible for or its place for enzymatic reaction, and so all the enzymatic reactions in chloroplast will occur in the stroma. It is also responsible for synthesizing sugar that in turn form the starch. So the sugar that is formed in this like this chloroplast will be converted into starch. In case of plants, we know that the stored material, stored food in plants is in starch form. Okay, so okay. Okay, so what happened? The enzymatic reactions and this ATP and NADPH reactions, they are like directly dependent on the light. Therefore, these are known as light reactions or photochemical reactions, whereas sugar synthesis and this ATP, this starch, they are not dependent on the light. Like they are dependent on light, but it is not like that. Okay, they have to done in presence of light. Therefore, to distinguish between the light and dark reactions, they have given them another name, dark reaction, that involves carbon reactions or synthesis of carbon. So dark reaction doesn't means that they will occur in night or darkness. Okay, they just to distinguish different process that are occurring in the chloroplast, they have given them two different names, like light reactions and dark reactions. Okay, बेटू अनवद्या समझ में आ रहा है ना बेटा?
Yes, ma'am.
Asmi, Rishad, good beta. Should I proceed?
Yes, ma'am. Yeah.
Okay, so basically, so this thing you have to remember, and I think question can be asked on this. The process of photosynthesis takes place in which part of leaf? Then you have to say it will take place in the mesophyll cells of the leaves inside the chloroplast. And the chloroplast consists of many disc-shaped structures known as thylakoid that consist of the pigment known as chlorophyll. So the chlorophyll is basically responsible for absorbing certain portions of visible spectrum and captures energy from sunlight, jahan pe this process of photosynthesis will occur with the help of chloroplast, because chloroplast will be able to absorb some portions of light, like visible spectrum, that is responsible for process of synthesis, photosynthesis. ठीक है?
Yes, ma'am.
Like समझ में आ रहा है ना? ऐसा कुछ confusion जैसा नहीं है ना? Should I proceed?
Yes, ma'am.
अच्छा, all of you have submitted the test copies, answer sheets?
Yeah, ma'am.
Yes,
Okay.
ma'am.
अनवद्या ने तो जस्ट अभी अनवद्या ने शायद अभी सबमिट किया है. ठीक है, चलो. सो सैटरडे को टेस्ट ले लूं क्या एक और?
We already have a test coming.
That remaining chapter, which chapter
Ma'am,
number?
we have a chemistry test on Saturday.
Yeah.
So
Sunday. Yeah.
मंडे को ले लूं? ट्यूसडे को तो क्लास है.
Can you take it
Wednesday.
after Tuesday? Yeah, Wednesday.
Wednesday will be fine.
Okay.
Because we have our English project also. So we
Okay,
are
fine,
supposed to
fine.
submit it by Tuesday.
Okay, I will take it on Wednesday. And Risha, please send me that thing, that details, है ना, so that I can
Yeah.
plan accordingly. First, I should finish your syllabus. Then the remaining
And
portion.
we have 4 more chapters left plus, yeah. So I guess we can finish it.
But I have to take test as well. Objective test for all the chapters. Subjective test I think I have taken for almost all the chapters, but objective is still remaining, no? So I have to take that also. Okay. Okay. So how many types of pigments? There are a lot of pigments other than chlorophyll that is involved in photosynthesis. So how can we come to know that what are all the what are all the pigments that are involved in process of photosynthesis? So a chromatographic technique, chromatographic separation. Chromatography is a method or a technique that helps in getting like different, it helps in separation of different pigments present in anything, or we can say present in leaves. So by chromatographic separation we come to know that there are 4 pigment other than chlorophyll that are involved in process of photosynthesis. So they are named as chlorophyll A, that is that is bright green or blue green in appearance. Chlorophyll B, it is yellowish green. Xanthophyll will be yellow, and carotenoids are yellow to yellow orange in color. So what are pigments? What is the definition for pigments? Pigments are the substances that are capable to absorb lights at specific wavelengths so that they will appear different color. So you know that the colors of like rainbow um, are explained with a single term, Vibgyor, that represents the different shades present in different shades or different colors present in a rainbow. Okay. Now let us consider the diagrams. So in figure 13.3, we can see the wavelength or the color at which chlorophyll A shows maximum absorption. So this is figure 13.A. This blue color line represents chlorophyll A, yellow represents carotenoids, and green represents chlorophyll B. So you can see the peaks of chlorophyll A are at 2 places. Can you see? And they are quite away from each other, right? And the chlorophyll B is absorbing again at 2 places. One at one the peak is high, and another place the peak is quite low. Carotenoids also have 2 peaks, but they are quite closer to each other. Okay. So what does this graph means? This graph means in case of chlorophyll, the maximum absorption in red region, so there will be one red region and another will be the blue region. That so red region mein absorption will occur at 642 nm spectrum, okay, and the blue region absorbs 372 nm of light in spectrum, whereas chlorophyll B absorbs 626 nm of light and 392 nm respectively. They are the absorption spectrum or absorption peaks for chlorophyll A and chlorophyll B. So you have to remember these values. ठीक है? Red and blue light absorb करता है chlorophyll A, and this chlorophyll A is basically involved in the process of photosynthesis सबसे ज़्यादा, because it is absorbing red and blue light. Red and blue light region में absorb करता है इसीलिए. Okay, so this thing you have to learn. Okay, and the second graph represents what is second graph? Second graph represents action spectrum of photosynthesis. Okay, so you can see here the photosynthesis process, red chlorophyll A, B and carotenoids. You can see like its values are not given here. I think in the last graph it is given. This is 400 nm, 500, 600 and 700. So वो कौन सा chlorophyll A is absorbing at 642 and blue at 372. Okay, so we can say this is 700, so 642 is somewhere here, and that is 372. So 372 will be somewhere here before 400. Okay, so this is the peak for chlorophyll A. Now in case of chlorophyll B, where is chlorophyll B? Here it is. So यहाँ पे which number is there? What is the absorption of? It will absorb at 622, 626 and 392. Okay, so 626, where it is 600. This is 600. You can see it is absorbing at here. So it is between 600 to 700. That will be 626, and another is 300. What? 392. Okay, so this is 400, and the peak starts from here. So it is somewhere 390. No, it is somewhere 392. 400 से यहाँ से start हुई है. Okay, so the diagram, I don't think you have to remember. You just have to remember the like numbers, because this is not very good diagram. So what we conclude from this is, okay, the maximum, the conclusion for this diagram is that maximum amount of photosynthesis occurs in presence of, like in red light. So light is absorbed by there are two photosystems, I will tell you later on. So the light is absorbed by photosystem second, that is also present on the thylakoid membrane. It absorbs the infrared and red light. This, just remember this thing. So what are carotenoids if a question is asked? So the carotenoids are the phytonutrients. What are phytonutrients? Plant chemicals or the nutrients that are obtained from plants. They are found in the cells of wide variety of plants, algae and bacteria. What is their function? They help in plants to absorb the light energy for use to be used in photosynthesis process. Okay, so the conclusion is that chlorophyll A is a chief pigment that is associated with photosynthesis, and the maximum absorption by chlorophyll A is done in red and blue spectrum. ठीक है? So basically the chlorophyll is the major pigment that is responsible for the network problem is from my side or from your side. I don't know. Risha and Anavadya.
I got disconnected.
Okay, fine. So where we were? Okay. So chlorophyll is the pigment that is basically involved in trapping of light. Other thylakoid pigments if it is asked that out of the following, which is the major pigment that is responsible for light trapping in case of photosynthesis, and if the options are given, chlorophyll, carotenoids, xanthophyll, and chlorophyll B, chlorophyll A or chlorophyll B, then you have to answer chlorophyll A is responsible. And these chlorophyll B, xanthophylls, and carotenoids, they are accessory pigments. They are also responsible for absorption of light, but they are not very much able to absorb light in comparison to chlorophyll A. So they also absorb little quantity, small quantity of light, and what they did, they transferred the energy to chlorophyll A. Okay. So if a question is asked, write the major pigment as well as the accessory pigments involved in the process of photosynthesis or for the absorption of light, then you have to answer chlorophyll A is the major pigment, whereas chlorophyll B, carotenoids, and xanthophylls are the accessory pigments. They also absorb small amount of light and they transfer their energy to chlorophyll A. Okay? Or what are all the other components that helps chlorophyll A in photosynthesis or light trapping? Then also the answer will be same.
Ma'am,
Beta.
ma'am, leaves are green because they reflect green light, no?
Because of presence of chlorophyll, beta.
Yes, ma'am, but if
Hmm.
they absorb red and blue, that means they reflect green, and that's why they are green in color, right?
Green in color, because that chlorophyll jo hai na, that because of this red and green light absorption, the chlorophyll reflects its green color, or the green color of chlorophyll is because of that only. उनका light absorption red and green में होता है, इसीलिए they appear green.
They absorb red and blue.
Red and blue, sorry. And
then why are they green?
It is like that only. There must be some concept behind this, but I don't know the exact concept. I know
Why
that
do they then absorb red and blue? Like, what is the use of absorbing red and blue?
particular wavelength is responsible for, like, required for photosynthesis process to occur. तो रेड एंड ब्लू लाइट भी जो पर्टिकुलर एनएम दिए हुए हैं ना स्पेक्ट्रम के, उसी एनएम में एब्जॉर्ब करेगी, देन ओनली द फोटोसिंथेसिस will occur. This is the internal mechanism of plants, that they will absorb the particular amount of red and blue light at particular spectrum or particular wavelength, then only they will be able to uh, involved in photosynthesis. ठीक है ऋषा?
Yes, ma'am.
So I told you earlier also, there are two types of process.
Ma'am, but then they need green color for their photosynthesis. No, that's their requirement.
They don't need green color, they need chlorophyll.
Yeah, ma'am, but chlorophyll is green.
Yeah. So, tell me what you want to ask. Ask. Risha. I think she got disconnected again. Okay, so what is light reaction? Light reaction are also known as photochemical reaction, that occurs in presence of light, the chemical reactions that occur in presence of light. This include light absorption. So photochemical reaction or light reactions uh, are like doing many functions. Some of them are absorption of light, first of all, then second is splitting of water, third is release of oxygen, and fourth is formation of high energy chemical intermediates like ATP and NADPH. So these are all the steps that are involved in process of chemical reaction, or we can say light reaction. This thing you have to remember, the steps for light reaction, okay, or the things that are produced or synthesized or included in the light reactions. So there are many complexes of protein that are involved in this process. So the pigments are organized into two discrete photochemical light harvesting complexes within the photosystem 1 and photosystem 2. So there are two light. What is light harvesting complex? So the complex structure that involve many things for harvesting light is known as light harvesting complex. And this light harvesting complex is made up of 100 type of pigment molecules that are bound to protein. So these are the pigment molecules. They are somewhere bound to proteins. So there are two photosystems, photosystem 1 and photosystem 2. Each photosystem has all the pigments except one molecule of chlorophyll A, because this chlorophyll A molecule forms the antenna of the light harvesting system. So this reaction center is made up of chlorophyll A, and these all are the other pigment molecules. So light in form of photon will come and is trapped by these pigment molecules. Okay. ठीक है. So okay, so this is the structure of light harvesting complex. Again, I'm repeating, there are many pigment molecules that will absorb the light energy in form of photon. This is reaction center, that is chlorophyll A. So light will fall on these pigments and the electrons will move towards the primary acceptor, that is primary acceptor definition I have added somewhere. 1 minute. I don't know, somewhere I have added what is primary, like what do you mean by primary acceptor? If I'll get, and like in between I will tell you. Okay, so this is the these are the different portions of light harvesting complex. It is clear?
Yes, ma'am.
Asmi again got disconnected. No, sorry,
No,
Risha.
ma'am.
Risha. There is some network error at her side. ठीक है, Anvadya, understood beta?
Yes, ma'am. Yeah.
So this, what happened nowadays? The biology class is getting boring. And you both of you are sounding very low. What happened? Health is good? Asmi is doing studies nowadays. I can see dark circles under her eyes. Good beta, you should do study. And I will check copies, test copies, then I will come to know who all are doing studies and who are not. But I think you all are doing. Acha hai, because the exams are coming and you should do. Okay, so the light harvesting complex or antenna, or it's an array of protein and chlorophyll molecules. So यहाँ पे yes. So here the acceptor will be what? The acceptor will be, we can say it's an antenna, or chlorophyll are the pigment molecules, or the reaction center is chlorophyll A. They are embedded in the thylakoid membrane of plants. So you remember what is thylakoid? It's the structure that is present in the chloroplast.
Oh, yeah.
Yeah. And they are responsible for transferring light energy to one chlorophyll A molecule that is present at the reaction center of photosynthesis. Like this, what we can say, photo system. So what will happen? These are the pigment molecules. This is chlorophyll A reaction center. This is light. Light will fall on this. They will trap the light energy and they will transfer it to this reaction center. Then these reaction centers से जो आपका light या electrons आए, they will get excited and they will be accepted by primary acceptor. ये I will explain you with another diagram in this class. So what is the main purpose of these pigments is these pigment helps to make the photosynthesis more efficient by absorbing wavelengths of light. I told you earlier that chlorophyll A is the accessory pigment, most important or primary pigment that is involved in process of photosynthesis, and there are many accessory pigments like carotenoids, chlorophyll B, and xanthophyll. So these carotenoids, xanthophylls, and chlorophyll B are the other pigments other than chlorophyll A. So these pigments help make photosynthesis more efficient because they absorb different wavelength of light. So the single chlorophyll A molecule form the reaction center. As I told you earlier, this particular thing represents chlorophyll A, that is a reaction center. Okay. So there will be a difference in the reaction center in case of both photosystem. In case of photosystem 1, the reaction center will be chlorophyll A and it absorbs the peak at 700 nm. So it is known as P700. Photosystem 1 is also known as P700. Why? Because here chlorophyll A absorbs the light at 700 nm. Okay. And photosystem 2 is known as P680. Why? Because it absorbs maximum at 680 nm. So this thing you have to remember. A question is asked generally that why photosystem 1 is known as 700 and photosystem 2 is known as P680, because they absorb the 1 minute. I'm coming 1 minute. 1 minute, huh? Don't shout, keep quiet. Okay. So I was not telling you, okay? Because you was shocked. Okay, I'm not shouting why ma'am is saying like that. Okay. So it's clear what's the difference between P7, like why photosystem 1 is known as P700 and photosystem 2 is known as 680. It's clear, Anvadya, asked me? Okay.
Yes, ma'am.
Yes, ma'am.
So as like the reaction center is different in photosystem 1 and photosystem 2. Similarly, the electron acceptor is also different in photosystem 1 and photosystem 2. So in case of photosystem 1, ferredoxin act as primary electron acceptor in photosystem 1, whereas in case of photosystem 2, pheophytin act as electron acceptor. They are the name of the pigments or ferredoxin, like they are responsible for absorption of electrons or electron acceptors. Okay. Now we are going to study the most important and most interesting topic of this chapter, the electron transport. Okay. So let's see this diagram first. What will happen? The light will come and fall on this light harvesting complex. Then it will, like electrons or get excited. Then the electrons are excited to much extent so that they can reach to electron acceptor. From here, these electrons are passed to electrons transport system, from where these electrons are transported to light harvesting complex of photosystem 1, and again the electrons get excited and are accepted by the electron acceptor of photosystem 1, that is pheophytin. I think I told you, no? Pheophytin, yes. Pheophytin will accept it, and it is responsible for reduction of NADP to NADPH. This is the schematic explanation. Theory I will explain you now. But the most important thing about this Z scheme is that why it is named as Z scheme, because we can see it is, this reaction is occurring in this manner, like it's forming Z. That's why it's known as Z scheme. And a question may arise, or you, any one of you should have ask me a question, that if the electrons from photosystem 2 are transferred to photosystem 1, then what about the electron loss that is caused to photosystem 2? How it is we can say neutralized, or how it is fulfilled? Then the electrons that are lost by photosystem 2 are regained by photolysis of water that will occur at the photosystem 2. So the water get hydrolyzed to form 2 electrons, 2 protons and oxygen. So this is the oxygen that we are using for process of breathing. Okay. And these electrons goes back to light harvesting complex of photosystem 2 to overcome the loss of electrons that they have transferred to photosystem 1. Okay, so now let's concentrate on the theory. So what are photosystems? Photosystem 1 and photosystem 2, they are the functional unit for photosynthesis. They are defined by a particular pigment organization and associated panel whose work is absorption and transfer of light. So they are two. इनके पास दो लाइट हार्वेस्टिंग कॉम्प्लेक्स होंगे, 2 इलेक्ट्रॉन एक्सेप्टर्स होंगे. So there are 2 types of photosystem, photosystem 1 and photosystem 2. Both photosystem consist of many pigments that help to collect the light energy, as well as a special pair of chlorophyll molecules found at the core reaction center. So what this light harvesting complex, as I told you earlier in that diagram, it consists of many pigments along with the reaction center. The reaction center will be chlorophyll A, and other pigments will be chlorophyll B, xanthophyll, and carotenoids. Okay. So now I will explain you the theory. In photosystem 2, the reaction center, that is chlorophyll A, it will absorb 680 nm of light, red light, that causes electrons to become excited. So here light harvesting complex mein this chlorophyll A, that is the reaction center of photosystem 2, will absorb red light at 680 nm, as a result of which the electrons become excited and will jump into the orbit that is quite away from atomic nucleus. And so these electrons are accepted by the electron acceptor, that will be ferredoxin in case of the photosystem. Photosystem 2 mein it will be pheophytin, I think. I'm getting confused. Yeah. In case of photosystem 2, it is pheophytin, and ferredoxin is in case of photosystem 1. So chlorophyll A will absorb red light at 680 nm, and the electrons are excited and the electrons are accepted by the electron acceptor. Now what happens? The electron acceptor will passes the electrons to the electron transport system. Okay. This electron transport system consists of cytochromes. This, I think, I don't know. Okay, this electron transport system you will be going to study in this chapter or in some other chapter, but I think this will be in your syllabus. So this electron transport system consists of different type of cytochromes. Okay. Now the electrons are not used in here in this electron transport system, but they are passed to the electron, they are passed to the pigments of photosystem 1. Now the reaction center, the electrons in the reaction center of photosystem 1, they also get excited and they receive red light at the wavelength 700 nm, and the electrons are again sent to electron acceptor. Okay. So this electron acceptor molecule consists of a greater redox potential. So what do you mean by redox potential? I think I have added. So Reduction potential or redox potential means the tendency of a given chemical species to be reduced. So reduce means, reduction means gain of electrons. Okay, so this electron acceptor, that is I think ferredoxin, in case of photosynthesis is capable to get reduced. So what is it did is, it is responsible for reduction of NADP to NADPH. ठीक है, रिडक्शन हो रहा है यहाँ पे NADP का NADPH में because of this electron acceptor of photosystem 1. So yeah, so these electrons then are moved downhill again. At this time, a molecule of energy rich NADP will be converted into, or it will reduce to NADPH plus H plus. Here H plus is not written in this diagram, it should be here.
What's NADPH?
Its full form is I don't know, but it's an like ATP की तरह ये भी एक source of energy है. I think 3 NADPH is equals to 1 ATP, or 3 ATP is equals to 1 NADPH. I don't know. I have quite confusion. I have not studied about that, but I will tell you. This is also an like energy rich compound. जैसे ATP है ना, वैसे ही. ठीक है?
Yes, ma'am.
Just give me 1 minute. I will tell you right now. बाद में नहीं याद रहता है. Full form of NADPH I will tell you. Yeah, it is nicotinamide adenine dinucleotide phosphate, or it is a cofactor that is used in anabolic reactions such as Calvin cycle and lipid and nucleic acid synthesis. So this NADP ka energy I will see. NADP energy. Yeah, so 3 ATP, yes. Okay. So 1 NADPH is equals to 2.5 ATP. Yes. So if 2.5 ATP are combined, then 1 NADH will be formed. Okay, so no, NADH and NADP are different. 1 minute, huh? I am searching about NADH. This is NADP. NADP. I will share a video with you, then this doubt will be clear. Okay, I will share a video
Yes, ma'am.
of this light Z scheme. I have seen on like YouTube. I forgot why this NADP is in, why this NADP is involved. I will tell you, but for now you can like remember that this NADP, that is nicotinamide. What is nicotinamide di? And I again forgot the full form of this. Nicotinamide dinucleotide phosphate. So this it's an like we can say cofactor है ये, जिसको ये this photosystem is reducing. ये cofactor further when we'll study about different cycles, that is Kelvin cycle, and there are many cycles for different mechanisms occurring in the plant, that is used in that. This will be used in that. Okay, so this whole scheme of transfer of electron that starts from photosynthesis 2 and goes till photosystem 1 involves the excitation of electrons and the transfer to another acceptor, that is from photosystem 1 to photosystem 2. Then again the NADP is reduced to NADPH, and one proton is also released, or we can say formed, that it is occurring in a Z shape, so it is known as Z scheme. Okay, now what happened, अब जो इम्पोर्टेंट चीज पढ़ने की है, यहाँ के सारे इलेक्ट्रॉन्स यहाँ पे आ गए, तो यहाँ से जो लॉस हुआ इलेक्ट्रॉन्स का, हाउ दैट लॉस विल बी कवर्ड. सो व्हाट विल हैपन, what will happen, the electrons that were moved from photosystem second must be replaced. So this can be achieved by electrons available due to splitting of water. So what happened, the water is splitting is associated with the photosystem 2, where it is split into two proton, oxygen and electrons. This creates oxygen, that is like we are taking the oxygen from plants. In this way the water is the oxygen is formed. So one of the net products of photosynthesis, so what happens, two molecules of water are split to form 4 protons, 1 oxygen molecule and 4 electrons. ठीक है, so ये reaction कहाँ होगी, this reaction will occur in the thylakoid membrane में होगी ये रिएक्शन, जिसमें आपका water is split into hydrogen and oxygen is produced as a byproduct. So what happens, the chlorophyll molecule that is left without an electron can take an electron from water splitting. So here photosystem second में जो electron chlorophyll ने gain किए थे, they have transferred that to photosystem 1. So here the chlorophyll will be there but electrons will not be there. So जो thylakoid membrane पे water splitting से proton मिलेगा, sorry electron मिलेगा, this chlorophyll molecule in light harvesting complex of photosystem 2 will receive the electrons from the splitting of this water, so that the deficiency of electrons can be overcome. Okay, so hydrogen and okay, so as a result of which the oxygen is released into the air as a byproduct of photosynthesis. So the end point of light reaction, or what's the main aim of light reaction, is to make large quantity of NADPH and ATP. So what is this NADPH and ATP? They are the ATP is the energy and NADPH are the cofactors that will be required for further metabolic processes that will be occur in the plant. So I think I have prepared till this only. So Risha, just a minute, and now I will search your query. I have time. So what was your question, Risha?
Ma'am, what NADPH?
Yeah, that
So कहीं भी इसका particular answer I'm not getting. It is written here like that. Chloroplast in case of plants uses this NADPH. It's an like part of the pathway for synthesis of oxygen or their food, that is carbon dioxide or sugar. So this NADPH helps in carrying electrons and protons driven by sunlight to a new carbon or carbon bonding products.
Ma'am, I just asked the full form of NADPH.
So I told you that full form, no?
Yes, ma'am, that's only the doubt.
So I told you that full form, but like it's a part of like the process that is involved in formation of like photosynthesis or formation of food of plants. ठीक है? Nicotinamide diaminotide phosphate is its full form, I think. That is nicotinamide adenine dinucleotide phosphate. It's very tough. The chemical structure is also given on Google. So, ठीक है? So, ये जो ATP and this NADPH that are synthesized during the this Z scheme, they will be used during the Kelvin cycle and Kelvin cycle for fixation of carbon dioxide. So these NADPH and ATP that is produced during this process will be used in Kelvin cycle. That is a different cycle. One thing I forgot to tell you that यहाँ पे जब electrons are transferred to electron transport system, then ATP will also be formed. ठीक है? One ATP molecule, that is energy, will also be formed. ठीक है? So at the end of Z scheme, what we are gaining is one ATP molecule, one NADPH molecule, and two protons and oxygen molecule, because the electrons are used by the photosystem second. ठीक है? So I think a question can be asked at the end of Z scheme. Z scheme, what we get. So these are the things that we gain, at. So or one more question can also be asked, like which is the final acceptor in case of Z scheme. So you should say NADP is the final electron acceptor in case of Z scheme, क्योंकि ये electrons that are accepted by electron acceptor are transferred to NADP. That is just where the NADP is reduced to NADPH. So it is the final electron acceptor. And which is the first electron acceptor in this Z scheme? Light harvesting complex electrons are transferred to this electron acceptor of photosystem 2, that is pheophytene, and photosystem 1 ka electron acceptor is paradoxine. This thing is not given in your book, but you have to remember. क्वेश्चन पूछा जा सकता है कि फोटोसिस्टम 2 का इलेक्ट्रॉन एक्सेप्टर क्या होगा और 1 का क्या होगा. आई हैव एडेड इन द नोट्स and I have to like send you the notes for last chapter only. I think uske pehle ke notes I have transferred. I have like posted notes, or for which chapter I have to post the notes, I don't remember. I think I have to post notes for last chapter only.
Ma'am, should I check and tell?
हाँ बेटा, check and just drop a message in group or like जैसे भी. So this cyclic and non-cyclic photophosphorylation, I think we are going to study in next class. This is थोड़ा सा tough chapter है, but थोड़ा interesting भी है. It's complete in your school. Your ma'am completed this?
No, ma'am.
Okay. So this portion that I have taught you today
Ma'am,
is
you gave till chapter 19 notes.
okay. So chapter, I think chapter 15 or 13, yeah, 15 we have finished in last like on Monday. Today is what? Today's
Monday,
Thursday.
ma'am. Monday, yeah.
Yeah. So I will send you the notes of
Sorry,
the
Tuesday. Tuesday you finished the
yeah,
chapter.
Tuesday. So I will send you notes of that chapter right now. And test का क्या करें? ठीक है, Wednesday को I am going to take test of chapter 19. It's fine.
Yes, ma'am.
Okay. और chapter वो जो chapter like what 13 is this 13 15 chapter 15 का भी just tell me when you all are able to give test क्योंकि I have to take objective test as well. So subjective complete हो जाए तो थोड़ा objective लेने का time मिल जाएगा. Okay. So how much time is left? I think time is
20
left.
minutes. 20
20
minutes.
minutes. So if you have any doubts, you can ask me because I
And
have.
from the test, can be asked out?
Oh, चलो ठीक है. Ask. I have
Ma'am,
not.
test, you're sending the answer keys, right?
Yeah, I have to prepare the answer keys. I think most probably I will prepare the answer keys today. I am not getting time यार. इसलिए I have to prepare the answer key. ठीक है
Ma'am,
ऋषभ.
the two tests, like many questions have been repeated a number of times. Like 3 to 4 times they have been repeated, the same question.
In this like
Actually, in both the tests.
for 17 and 18.
Yeah.
अच्छा.
Yeah, ma'am.
Ma'am, and the last to
What
last
is the-
test, there were even
What is the KB number?
questions wrongly marked.
Yes,
Like it's
ma'am.
just a 1 mark question. It was marked like 3, 4 to 5 marks.
No, it's fine. It depends on me. Okay, how much marks I actually I have to prepare question paper for only 80 marks, I think, right? So I'm not getting very
Ma'am,
much.
and from the next paper, we won't be having MCQs.
Yeah, ma'am. Our new pattern, we don't have MCQ questions.
Okay, so what's the new pattern? You'll be
We
I
getting
don't
think
know.
regular like first 5 questions, 1 mark of 2 markers. I mean 1 markers, like fill in the blanks, and it's like that
No,
one
like not fill in the blanks, like proper questions will be
questions.
given.
Yeah, like
one
huge
line
fill
or
in the
one word
blanks.
questions.
Yes,
Yeah.
ma'am.
Okay.
Yeah, right.
So paper will be of 80 marks, right?
Ma'am,
Ma'am,
they don't have
60 marks.
Okay, so I
50, sorry. Our last midterm paper was 50 marks. I'm not sure how much our pre-board paper will be.
so pattern will be like that. I have to give few questions of 1 marks, few questions of 2 marks, and then few questions of 3 or 5 marks, like that.
Yeah,
Ma'am, we
ma'am.
don't know the marking like distribution because we might even have 5 and 6 mark questions.
But you told
But
me
ma'am,
not
yeah, they will be divided into sub questions.
parts. Yeah. So I will prepare actually for last 2 test papers. I have not got very much time, so I prepared them randomly. Jitna question mila utna because I was having my like house inauguration.
Because for blastosis futalis, I might have written the answer around 3 times, ma'am. The matlab, other questions were repeated.
Actually, I have taken question from somewhere, and I have like I was not having very much time to see the question paper even. I have just prepared the because it now as a Thana decided that questions we are not going to like take test right now. You all were not ready, but I have to take the test, so I took it. But if you want, then I can repeat the test for these chapters. But there was not any question problem in the questions. No, only repetition was.
Repetition and ma'am, 1 marker questions were like they were given. We had to write for 3 marks, but it wasn't possible because we only knew a 1 marker answer for it.
Oh, it's
There was very
fine.
limited information.
So don't worry about that. Don't worry about marking. It depends on me ke, because I was not having very much time as well as I was not having very much questions also, and I don't have time to search for more questions. So I use the questions as it is, and marking I have done it on my own. So I don't know which questions should have how many marks. I have just given randomly marking. So I will check accordingly and will give you marks accordingly. From now onwards, I am getting time and I will get time to prepare good paper and answer sheet as well. Okay. So Risha, once I prepare answer sheet, then you can, it will be fine if you will ask me questions then.
Yes, ma'am. Yeah.
You know, because I'm not having answer sheet right now, so how can I satisfy you? Okay, yes, this is the answer and how much you should have written. Even I have to check your copies as well. I will check. And for that biomolecules, I'm not going to check copy because I think I will teach you all that chapter once more and then I will take test. It is fine.
Yes, ma'am.
Okay, the biomolecules test, right?
Yeah. So I will be teaching once I'll finish your Risha, please send me the details so that the portion that is coming in your syllabus, I will finish that first and then we will do the
Ma'am, I
rest.
had sent you the revised syllabus, right? We have
No,
only 4 more chapters remaining.
yeah, I know. So I don't know, beta. It might be possible I have missed that. So is it possible for you to write the portions of each chapter that are included in your syllabus? So I will cover them first, then I will teach you rest portion of the chapter so that at least your syllabus will be complete. So I can teach
Ma'am, can I do one thing? Can I take that particular portion,
photo.
like
Huh.
screenshot it, crop
Yeah,
it, and
yeah,
then
sure,
send it to
sure.
you?
Send me like that only. No need to write. You can take screenshot or photo, whatever you want. You can send me that also. The portion that is included in your syllabus, I will cover them first so that it will be good for you and me both. Portion complete ho jayega, then we will start taking objective test and subjective test, and then the remaining portion I will keep on teaching you. Okay, so that I can plan accordingly that I have to take regular classes or the classes that I am taking right now are sufficient. I have to decide that. Okay.
Ma'am, do we have to? Will we be asked we will be asked the heart diagram, right?
I think I don't think so, but it depends on examiner, beta. We can't do anything. It depends on the mind of the person that is making paper. Usually, it should not be asked because it's quite like tough diagram.
Complicated, yeah.
Yeah, it's quite tough diagram,
But we have drawn heart diagram for many exams previously also.
Yeah, last year I think.
Yeah, from 8th standard we have got that heart diagram, so
yeah.
it might be asked.
So if it's possible for you to, but like I will say, okay, if it's possible, then try to learn the diagram because it's very interesting and useful too. But it depends on like you should learn and kar loge. It's you have to. You can't do anything because if you will not learn the diagram, then you will not be able to like understand different portions or utna theory yaad karna is quite difficult. If you remember the diagram, then it's easy.
Yes, ma'am.
Okay. ठीक है चलो अब anyone like more
Ma'am, I
doubts?
sent the portions.
Okay. Okay, so these TCA cycles and they will be in respiration part. Okay. Plant growth and development, growth regulation. Hey, this chapter is complete. Risha, we are done with chapter 15.
Oh, we are done? Then only 3 chapters because I was absent, so I don't know.
We are done with chapter 15. I finished the chapter on Tuesday only. I will be sending you the notes of that chapter right now only. Chapter 14,
Okay,
21 and 22. And including this chapter, we have to complete 4 chapters. So what I will do, I will start chapter 14 after this chapter because they are correlated. These both chapters are correlated. So after that, we will do these 2 chapters. ठीक है. ठीक है बेटा. So if there is any doubt from today's chapter, what we have done till now, then you can ask me. I will send you a video related to this Z scheme also. So it will be helpful for you to see.
Okay, ma'am.
Okay, then I'm done with today's class. We'll
Okay,
meet
ma'am.
you on Saturday. Okay?
Yes, ma'am. Thank you,
Okay.
ma'am.
Thank you, ma'am.
Bye. Have a nice day.
Same to you, ma'am.